Hello, good morning, good afternoon, good evening from wherever you're joining us. You're welcome to this lecture. I'm going to be discussing the development of the special senses with a focus on the eye. So I remain, my humble self is Jupo Kafo, and we're going to look at the embryology of the eye today. You're welcome once again. We're going to start this lecture by looking at the, overview of how everything gets started. Special senses, we have basically, when you are probably in your college, you're told that the special senses, the sense organs are five. But now when we talk about special senses, we're talking about organs that are designated for a specialized kind of sense. And there are just about four of them. The vision, that is for the eye, the gustation, olfaction, and audition. These are the four special senses. And before we talk about the eyes, which is responsible for vision, we want to know how everything comes to be. So the development of the face is a major part of the development of the head and neck, where you have the presence of all these senses being located: the eyes, the ears, the nose, the tongue. All these organs which are specialized for these senses are situated in the head and neck. And around the third week, towards the end of the fourth week, you have the appearance of the stomodium. I'm just going to uh, use my laser to put that up. So you can see the appearance of the stomodium, which is more like the primordium of the face. So this appears around the end of the fourth week. This must have been established to be able to situate, the appearance of the progenitors of some of these senses. So basically, you have the appearance of the mesenchymal prominences, from that time of the fourth week and then up until the seventh week. By the seventh week, the face would have been established to some extent, having at least some significant prominences. These are characteristically, five of them. You have two mandibular prominence, which you can see, using my laser pointer. You can see it on the right and on the left. And then you're going to see two maxillary as well, which you can also see, cranial to it, on the right and on the left as well. And then you can see the stomodium just between the two maxillary prominences. And then you see a frontonasal prominence. At this time, now you can also appreciate the nasal placoid, which is already upcoming, which is responsible for the formation of the nose as well. And then with time, you're also going to see the otic placoid, which will also be situated, just very close to the frontonasal prominence. So this establishes the beginning of the formation of these senses. And as you monitor the embryo growth, these are the signs that is being seen, especially with MRI or with ultrasound, to be able to monitor the growth of the baby. So further differentiations that will begin to happen from this stage gets to establish the specificities that are peculiar to any particular sense organ. So now it's very important to also remind you that all these sense organs, develop simultaneously. The eye, for example, begins also in the third week. So there would have been some signs that something is happening, especially mobilizing the cells that are responsible for this. And this process begins from third week. I made mention of the establishment of the stomodium, which is the primordium of the face, around the end of the seventh week. So you can see that the eye, starts almost at the same time when these prominences are appearing, and also continues even beyond the establishment of the face. Characteristically, it's important to note that ectoderm and mesoderm actually make contributions to the formation of the eye. So you have components which are coming from the ectoderm, and you have components that are coming from the mesoderm, especially for the fact that the eye, as we all know, is responsible for sight, but then you have some muscular components that regulate. You have some, muscles. Even sometimes you have, contributions from the facial muscles, to regulate the movement of, the eyeball, for example. So you also have some intra and extraocular muscles that are responsible, for movement of the eyes, regulation of how, the eye performs its function, especially things like accommodation and, regulation of the light that comes into the eyes. So that will tell you why mesoderm, which is usually responsible for formation of these muscles, play a role in the formation of the eyes. Now, I try to make it simple. For the ectoderm, we have the neuroectoderm, we have the surface ectoderm, we have the neural crest cells, and then we have the mesoderm, which sends its own cells that make contribution, especially for formation of some of the muscular cells. So we're going to be seeing how these components contribute at each point to lead to the formation of the eyes. Now we're going to see how optic grooves. If you look at this picture, you can see the appearance of the optic groove. Usually this appearance is around what we call the optic sulcus, which appears usually when the neurogenesis has started. Neurogenesis is the development of the brain. So the formation of neuropores, the cranial and caudal neuropores, which are initial signs of the formation of the brain and the spinal cord, comes up, and then you have the appearance of the brain into forebrain, the midbrain, and the hindbrain. So at the level of the forebrain, we are going to see the establishment of the brain into different divisions. For example, prosencephalon is being formed, and then that prosencephalon will lead to division of further two, which will lead to formation of diencephalon. At that the medial part of that diencephalon, you are going to see the establishment of an optic sulcus, which will eventually turn to the optic groove that we are just seeing. So this picture, you can see from this point, that seems to be a diverticulum or a depression that was shaping the entire tube, the entire cranial tube that is about forming, to take a shape. This shape, getting it to form something that is groove-like in nature. So the appearance of this optic groove is the major marker of the beginning of the formation of the eyes. It's important to note that at the beginning of the early stages of the development of the eye, two things are very important. The formation of the optic groove, or initially the optic sulcus, and then the lens placode. While the optic sulcus, emerges, or optic groove eventually emerges from the neuroectoderm, the lens placode actually, gets to emerge from the surface ectoderm. Okay, the lens placode. So I've tried to give the two pathways, and again, it's important to know that the induction of the development of the optic groove also leads to the formation of the lens placode. So you can see, again, let me pick my pointer. So you can see here the formation of the optic groove. Let me start with this point. Here you're going to see the forebrain. This is the cavity of the forebrain at this point. And then you are seeing the optic sulcus, which I said is just, an impression. And then this impression begins to grow towards formation of a vesicle. And then you can see from that neuroectoderm, an emergence of somewhat of a vesicular shape begins to form. And then it forms up to the point that it produces the optic vessel. This is usually as a result of some level of induction by the surrounding mesenchyme. So this induction by the surrounding mesenchyme is also in response to the neural folds. When the neural folds, at the cranial and the caudal begins to fuse, it leads to these inductions. And these inductions causes the neural groove, which is already formed here. You can see the groove, and then emerging to produce the optic vesicle. And then the more elongation of that vesicle will lead to the formation of the optic stalk, which is what you can see right here on both sides. So now we have the optic vesicle, we have the optic stalk, which is a formation of the optic sulcus and the optic grooves. Now let's go to the next picture you can see right here on the page. So now the optic vesicle, remember that there are also surface ectoderm surrounding the entire neuroectoderm. We just picked a part of it to just show you how the process is being done. So now the appearance of the surface ectoderm, which you are seeing. So the surface ectoderm is actually going to now start responding to the presence of the optic vesicle, and of course the changes will continue. This usually happens towards the ending of the 3rd week, towards the beginning of the 4th week. And then you have the surface ectoderm mobilizing cells, of course, from the surface ectoderm, the cells of the surface ectoderm emerging to form a placode called the lens placode. The lens placode eventually is going to lead to the formation of the retina, but let's look at what happens to it. So the lens placode begins to invaginate. So it is actually the lens placode that begins to invaginate towards the optic vesicle. And then you can see as it begins to invaginate, the shape begins to change, trying to invaginate towards the vesicle. As it does that, the vesicle will begin to change its shape towards a cup-like nature. And then while the shape of the surface ectoderm also begins to take the seating of that cup, as you can see right here. So as this is going on, you now see that the lens placode itself, the two parts, the upper and the lower parts, will begin to touch each other, and at a point they meet each other and break off from the surface mesoderm, which you are seeing in the last picture. And by that time, the optic vesicle would have formed an optic cup, while the pinched off part from the surface mesoderm becomes the lens vesicle. Now it becomes, because of the vesicular shape, it changes its name from lens placode to lens vesicle, while the surface ectoderm retains its initial shape back to the surface ectoderm. So there is a disattachment, from detachment of the initial surface ectoderm from the rest of the surface ectoderm to form the lens vesicle, which will eventually lead to the formation of the retina. So this is a typical early morphogenetic stage showing you, how the neuroectoderm leads to the formation of the optic cup from the optic sulcus, and how the lens placode leads to the formation of the lens vesicle, which will eventually lead to the formation of the retina. So this is just to show you a bit of a clearer process of what we've been saying so far. So you can see here, notochord appearing, so you can appreciate what we've been talking about. You can see the 3 layers, the ectoderm here, you can see the mesoderm, you can see the neural plate, and then you can see the neural folding at this point, and then you can see the emergence of the neural groove, and then here you can see the tube beginning to grow even more, and then you can see the surface ectoderm line just, cranial to that, and then you can see how the brain, the initial process of the formation of the brain. So this happens alongside the development of the eyes, and this leads to further induction into, for example, the optic sulcus and the optic groove, just like we have explained. And then you can see what happens when you look at it, when you look at the whole structure altogether, the whole developing brain. So the brain has, at this point, has been divided into different sections, the telencephalon, the diencephalon, the mesencephalon, and so on. Now, you can appreciate that all we have been talking about is what is happening at the diencephalon, this second stage. So you can see that small tip at that point is actually the sulcus, the optic sulcus, which talked about that we eventually form the optic vesicle. So that is it here, optic sulcus. So this is exactly where we are talking about, where we're looking at, and then you have it being magnified here. You can see the surface mesoderm You can see the forebrain within it at the central core of it, and then you can see the sulcus. So this sulcus will begin to grow to this point, to this particular image, and then becomes a vesicle. Okay, the stalk, as we saw in the last page, and then the vesicle, and then as it keeps growing, the vesicle becomes even more until it gets the shape of the cup, as we can see on the C part right here. So from this point where, formation of the vesicle at this point, and then as it grows towards the surface mesoderm, it becomes the lens placode, and the lens placode invaginates into it on this stage until the cup is formed on this stage. Now the cup is formed, now we can appreciate the lens vesicle, and we can also see the retention of the remaining part of the surface mesoderm, and that process continues. Now we're going to see further how retina in particular grows, but this picture even shows further growth of other cells that are part and parcel of the eye. The eye basically have specialized cells, for example, the cone cells, the amacrine cells, the red cells, the regular cells, and so on. So these cells are very important for the specialized function of sight, regulation of light, accommodation, and so on. So these are also implicated here, and the colors will tell you which is doing what, where which part of the eyes is responsible for the development of these cells. So we're going to see that further as we go, but this is just to show you a summary of how everything starts from its relationship to the neural fold development, the development of the brain, especially in the diencephalon in particular, and then how it turns down to the last part where we have reached now, which is development of the lens vesicle. Let's look more closely at how the retina and the optic nerve, begins to form. We've been able to establish the fact that there is an invagination process, of the optic vesicle, and that invagination process, which forms the optic cup, right? And then the retina emerges from that optic vesicle that is invaginating. So as you can see rightly here, you can see the invagination process. Here at this point we have this invagination process happening from the surface ectoderm on the lens placode, and then at the end of the day, retina has emerged. Okay, retina has emerged, but that invagination process creates two different types of surfaces, or two different layers, an inner and an outer layer within the optic vesicle. As you can see within this emerging optic cup, that there is a space called the intraretinal space. Can you see that space right here? Okay, let me use my cursor. Okay, can you see that space right here? So this is the intraretinal space, that you can see in the optic vesicle. So this space right here is a division as a result of the invagination process of the optic vesicle. So you have the outer layer, you have the inner layer of the optic cup. So these layers actually eventually they are going to merge together, they are going to adjoin to each other, they are going to obliterate. So the space is going to be closed up, as you can see in the later picture. But before then, you are going to appreciate the fact that the outer space, the outer part, let me use this picture now to also show that as well. So you can see the intraretinal space, you can see the potential retina, the retina, lens vesicle here forming, which is the future retina, right? And then in this space you can appreciate a little bit more the outer space. This is the already invaginated, this is the optic cup now, the outer layer, then you can see the inner layer, and you can see the intraretinal space. Then within here you're going to, this is where also you have the retina studio, but it's not showing in this picture. So the outer layer is actually pigmented in nature. It has pigments or pigment granules, or you can say pigmented cells. And then you have the inner layer that differentiates into the neural retina. So this neural retina is made up of some of the specialized cells, the rod cells, the cone cells. These are the light sensing cells of the retina. They are made from the neural retina, which is as a result of differentiation of the inner layer. So this is just to show the layer of the retina, and eventually, if with more growth, you're going to have other presence of other cells within the inner layer of the retina in particular. Now, cone cells are here, red cells are here. You can see, horizontal cells, amacrine cells, all the ganglion cells and so on. So you can see all the in a full mature, you can have up to about 10 layers, including the pigmented epithelium pigmented cells, which is in the outer layer of the retina. So this is how you are going to have the formation of the retina. When it is fully formed, it's going to have the presence of all these cells, and you're having retina already fully formed. But something gets to happen even after that. Now, the axons from this retina, remember that you are going to have the presence of axons that are emerging from the neural retina. Remember, we said that neural retina is a merge off from the inner layer, so that forms the neural retina. So once the neural retina is formed, you're going to have the emergence of several axons that now project themselves into the optic stalk. This is the optic stalk. So it is projecting itself because that optic stalk will eventually close up and fuse and close up the lumen, and that will lead to the emergence of the optic nerve. So optic nerve is formed basically from by the axons that emerge from the neural retina into the optic stalk. So the optic stalk eventually becomes, gets obliterated, the lumen gets obliterated and becomes the optic nerve. So when this optic nerve is formed, you can also notice that there is an emergence of a groove, along the ventral surface of the optic cord, cup and the stalk. Let me use this picture to show that. So you can see this groove that is emerging. This groove emerges and is called the choroid fissure. You can also call it the retinal fissure. So this choroid fissure you can see here leads to the formation of, or rather allows for the passage of hyaloid arteries and veins. So these two hyaloid arteries, which you can see in the next picture here, you can see how it starts to make contact, to the retina itself. So the hyaloid vein, the hyaloid artery pass through the retinal fissure or the choroid fissure, and they are actually entrapped into the optic nerve. They are entrapped into the cup through which they are passing, and they fuse with it. Eventually, these veins, the vessels, both the artery and the vein, are going to eventually, die off, or more like obliterate and become a canal. They will actually form what we call the hyaloid canal. They normally will initially be supplying the lens which is still developing, and then they degenerate in the vitreous humor or the vitreous body, but then they leave a remnant that is called the hyaloid canal. Of course, I can't show that on those pictures here, but it's just for you to know that eventually the hyaloid vesicles become entrapped in the optic nerve, and they further, become, central retinal artery and vein, which supplies the lens, and eventually degenerate and then leave off a remnant that is called the hyaloid canal. So this is just a typical overview of how the retina and the optic nerve, develops. Look at how the iris and the ciliary body is formed. Now, this picture shows you, all I've been saying so far, at least at the point that we have explained. We can find here the lens vesicle, which eventually becomes a lens. We can also see the division of the outer layer and the inner layer of the optic cup, and then you can see the intraretinal space, which eventually gets to close up, and this eventually would become, in adult brain, as you can see in this model, become, the retina. And then you can see the inner part of the retina being labeled as the neural retina. And of course, you can see the optic stalk here, which eventually becomes the optic nerve. And then initially in the embryo, you're going to have the hyaloid, but at this point, the hyaloid would have disappeared, because you would have had the presence of choroid plexus, and retinal arteries, would have emerged as well. Now, what happens that leads to the formation of the iris and the ciliary body, at the outermost part, the anteriormost tip of the optic cup, as you can see here. So you are going to see the growth of the optic cup towards the ventral location of the pupil, which is supposed to be just in front of the lens, right? So towards that part, you're going to see the emergence of a structure, the structure of the iris. So that's how iris is formed. So you're going to see it at the anteriormost tip of the optic cup. So you can see that on both sides, the iris, labeled here iris. You can also see that from the structures. But then when you look at the iris basically have two layers: the external layer that is pigmented, and then you have an unpigmented layer, and also you have other layers of pupillary muscles because of the presence of the two major muscles, the dilator, the sphincter. So you can appreciate that in this picture, dilator and the sphincter muscles, they are all pupillary muscles that are aligned to the iris. And iris also, have the inner wall of the optic cup, gives rise to a posterior iris pigment epithelium. While the inner wall gives rise to the posterior iris pigment epithelium, the outer wall gives rise to the anterior pigment epithelium. These are the two types of epithelium that you're going to appreciate, in the iris. And again, you are going to also see that the ciliary body, which is also formed from the same point, and the anteriormost tip, as you can see in this picture, and the anteriormost tip, the ciliary body emerges, is also formed at the anteriormost tip of the optic cup and has its own epithelium called the ciliary epithelium as well. And this epithelium will secrete what we call the fibrillins. Fibrillin is a kind of a protein or a molecule that contributes to the development of the suspensory ligaments of the lens. Remember, the idea is, trying to, be an anchor to this lens. So you can see that both of them, the iris and ciliary body, are all anchored at that point together with the ciliary muscles, which is responsible for lens accommodation, also emerges at this point, from the mesenchymal cells. This is just to get a bit clearer on it again. The lens, we've all already known that we've been harping on it. The formation of the lens plaque, shows the beginning of the development of the lens. And then the lens plaque will begin to invaginate into the optic cup, and then after that we're going to have the emergence of the lens pit, and from the lens pit, the optic cup divides into two, having the inner and the outer layer, and then you're having the emergence of the lens vesicle. This lens vesicle eventually becomes the lens itself, and then leads to further development of the lens. And then you have the emergence of the hyaloid artery and vein, that comes to the retinal fissure, and then this fissure will eventually regress, and you have further development of the lens vesicle matures into the lens proper. Usually what changes is the presence of the columnar cells. Okay, so the tall columnar cells on the posterior aspect of the lens vesicle, they lose their nuclei and then they lengthen, and then they form the primary lens fibers. And when this begins to happen, then you're going to have the emergence of the lens, in an adult nature. And then by that time, you would have had the disappearance of the hyaloid artery. So what is important, for lens is that lens is important for you to know that lens is an immune privileged organ. This is simply because of the presence of a lens capsule, which is usually formed. Unfortunately, this is not captured in any of the pictures, but the capsule is that it is encapsulated. It is within a capsule. So the presence of a lens capsule is usually formed, because it forms as a result of the same invagination from the surface ectoderm through which the lens plaque will emerges from. So you have the presence of a lens capsule, which makes it an immune privileged organ because you have that basement membrane, which has a lot of antipathogenic antipathogenic molecules present in them, surrounding, the lens at that point. So the capsule offers a pathogenic protection while still allowing things like water or even other solids to pass through the lens as well. So this is important for you to note in the development of the lens. Look at further development that happens in the eye, you're going to find out that there is what we call the vitreous body. This vitreous body is actually a network. It serves to maintain the shape of the globe, of the eye globe, as well as to secure the retina in place. So I'm going to have it, you can see the vitreous here. It's basically made up of fibrils, a network of delicate cytoplasmic processes which occupy the space between the lens vesicle and the inner layer of the cup. So you're having the lens here, then you're having the cup, right? So you're going to have that inner space that is left after the emergence of the outer and inner layer of the cup. You're going to have that space being intruded, usually as a result of the intrusion or invasion of the optic cup through the retinal fissures. So you're going to have mesenchyme invading the optic cup through the retinal fissure. The same fissure through which you're having the hyaloid arteries coming. The same fissure is a fissure that the mesenchyme invade through and form what we call the vitreous body. So we have, three developmental models. We have the primary vitreous, which develops around the first month of gestation within the first 4 weeks. We have the secondary vitreous, around the second month, and then we have further development in an adult, eyes, what we can call the tertiary vitreous, and then at this stage of the primary vitreous, you're going to have, cytoplasmic processes. At this stage, it's actually vascularized at this point. You're still having the presence of hyaloid artery. At that point, hyaloid artery has not, regressed, and then you're having it being supplied. So you're having mixture of, ectodermal cells, but majorly you're having mesenchymal cells, from the mesoderm, that constitute the primary vitreous. And then you're having the vessels, supplying them, as you can see in the second picture. Then we go further to the yeast maturation. One of the stages of that maturation to form the secondary vitreous is the presence of, or rather the regression of hyaloid, arteries, the vein and the artery. And then we are going to have the cell differentiate into what we call the hyocytes. They differentiate into a type 2 collagen cells, that has presence of hyaluronic acid as well. Then that sets the stage for the secondary vitreous body. In this case, the primary body loses its vasculature, then develops into some kind of an acellular gel, more like a jelly, substance. And then at that stage, we can say that is a secondary vitreous body. Then when it finally matures, it gives rise to the suspension fibers of the lens, and then also remains avascular as well. And then at that point, it coincides with the regression of the hyaloid artery, and then you're having a vestigial structure that is normally called the hyaloid canal in the adult. So that is the complete development of the vitreous body. The formation of choroid and sclera, begin around the 6th, 7th, and sometimes even up to the 8th week of gestation period. In fact, by the 3rd month, which is about the 12th week, you would have had visibilities of some components of this structure, especially the choroid, appearing. Choroid is a plexus of a vascular plexus. You're going to have capillaries, formed within them, because it's responsible for supplying, especially the outer part of the retina. So it supplies the outer retina. And normally, we know that endoderm is usually implicated in formation of capillaries. But in this case, this is formed from the ectoderm Okay, so you have differentiation of the ectodermal cells, neural crest cells, especially the mesenchyme. The induction of the surrounding mesenchyme is actually what leads to surrounding mesenchyme, usually around the optic cup, induces the formation of choroid. Now, choroid by the 22nd week of development, you would have seen the full adult eye formation, the full choroid, having all its plexuses, as can be seen clearly here. So you can see the placement of the choroid. You're going to see that at the outer part of the cup, okay, it supplies the outer retina, okay, which the cup which informs the retina itself. Okay, now you're also seeing the closely allied sclera. The sclera is actually forming exteriorly to the choroid. You can see the relationship between the choroid and the sclera. Okay, the sclera contributes to the stromal layer of the cornea eventually, but the sclera is actually ectodermal in origin as well, and also responsible, more like the fibrous layer of the eye, is continuous with dura mater, usually dura mater of the brain, and you also have it being induced from the paraocular mesenchyme as well. So by the 5th month, you would have had a fully formed adult sclera in place. So this diagram also shows you, up to the sclera and the choroid, what actually forms what. So you can see that neural crest cells, as I earlier said, forms the choroid and also the sclera as well. Is the clear portion of the outer eye, and of course it allows the passage of light into the eyes and then provides a large component of refraction. So you can see that clearly here. So around the 5th week, so you have the presence of migration of mesenchymal cells. This is normally being described as waves of mesenchymal migration. So what happens is that there is an establishment of the epithelium, the endothelium, and the interior core. So there are it creates 3 layers of the cornea. Okay, 3 layers of the cornea. The first is the external, which is the corneal epithelium, and then which is usually derived from the surface ectoderm. So you're having migration of mesenchyme from the surface ectoderm, forming that. And then you have the internal parts in between the corneal epithelium and the endothelium. So this internal part is called the stroma, is the central part, and then you have the interior part, which is the endothelium. So between the endothelium and the epithelium, there lies a central stroma layer. These are all formed by mesenchyme. And then you can see here the cornea being described here, the clear, portion of the eye, and you can see that there is a contribution from the key here. There is a contribution of neural crest cells, mesenchyme from neural crest cells, and also mesenchyme, of course, migrating from the surface ectoderm, forming that structure. So around the 5th week, you would have seen the emergence of these structures, and then you are also going to see the presence of the keratocytes, which are usually formed, within the interior part of the endothelium. And then these keratocytes are the typical cells that will be seen appreciated, as you can see here within, the stroma of the cornea. That I also inform you that cornea is usually avascular and also being induced by the formation of the lens vesicle. Okay, let's look at the embryology of the eyelid. So this develops, from folds. There are usually appearance of folds, as you can see, ectodermal folds, from the surface ectoderm, and then you are seeing one above and you are seeing one below. So you can see the appearance of the lens here, and you can also appreciate how that fold begins to look for fusion. So there is enlargement first, and then there is somewhat fusion trying to occur here. And then as that happens, there is a space that is normally left, which is the formation of conjunctival sac. Okay, and then you also see its association with the lacrimal gland as well. So this explains the relationship between the three structures, lacrimal gland, playing its role, to situate itself, just right close to the eyelid, and then you're having the conjunctival sac as well that is closely associated with the eyelids as well. So you have a closed space which is formed in front of the cornea, which is called the conjunctival sac, and then you have fusion completed by the two ectodermal folds forming the eyelids by the third month. So by the third month, this process is actually completed. Then you have an appearance of a mesodermal mesenchyme during the second month, which actually forms the muscles of the lid and the tarsal plate. These muscles are responsible for opening and closing the eyelids themselves. Okay, they are responsible for doing that function. And then you have the lids remain fused and then separating between the fifth and the seventh months of development. And then the eyelashes, of course, will emerge, from that point of separation. And then you're going to have the glands, also develop the lacrimal glands, which are responsible for production of tears, right? When we cry, they are also produced from the surface ectoderm as well. More detailed description of the lacrimal gland development, you actually will notice that this development of the lacrimal gland is divided into 3 stages, and it normally begins around the 7th week. Okay, so you're going to see that the gland, normally positions itself in the superlateral position of the orbit. Okay, so they position itself in that position, as you can see here. So within the orbit, it takes that superlateral position, and then it is formed in 3 stages. One is what is called the glandular stage. This stage is basically the thickening, of the surrounding mesenchyme. The mesenchyme begins to thicken and then begins to condense, from the ectodermal region, right? And then the second stage, which is the bud stage. At this bud stage, you're having the appearance of epithelial bud, as I earlier showed you in the previous slide. So you're going to see here, this is somewhat already bit grown because you're having, this is more like a mature stage now. So you're having the buds, begin to appear, and after the appearance of series of buds, not just one bud. So these buds grow bilaterally from the superior fornix of the conjunctiva into the underlying mesenchyme, and then they begin to canalize. The bud begin to create some spaces within them, having presence of ducts, within them, and then after which, they get to the third stage, which is the maturity stage. At this stage, which usually begins around the 9th week, keeps happening up until the third month, after birth, when actually tear production, can start. So you discover that tear production usually starts around, the third month. Have you noticed that babies at the first month, second month, you don't see them shed tears, when they are crying? So that is a puzzle for you to solve. So you can find that out. So you discover that tear production doesn't happen until after the third month, after birth. Okay, so this is the typical state. But then it's important to also note that, there is a development of a muscle called the levator palpebrae superioris gland muscle. Is a muscle of that helps divide the gland, or rather, the gland divides the muscles into 2 parts, the orbital part and the palpebral part. So this muscle is useful in movement of the eyelids, especially when, the eyeball itself, is being used, or even in crying, for example. So when those motions are being undertaken, it plays its role. So following up with the presence of the superioris muscle, we talked about the levator palpebrae superioris muscle. It's actually one of the 7 extraocular muscles. I've already mentioned the ciliary muscles, the sphincter muscles, the dilator muscles, coming as a result of development of the cornea, the lens, because of the role it plays. You also see that, related to the iris as well. So the iris as well. So you see the relationship. So they are called intraocular, because they are within, the eye itself. Okay, so you have the intraocular muscles. They are usually soft, tissues. They are smooth muscles, and you see them ciliary muscles. They help in accommodation, sphincter pupillae, and dilator pupillae. Of course, from the word dilate, the dilator pupillae dilates, while the sphincter pupillae constricts the pupil. So that's their specific function. While the ciliary muscles is useful for accommodation. Okay, so we also have the 7 extraocular muscles. These 7 muscles, you have 4 recti muscles, 2 oblique muscles, and then you have 1 levatopalpebral superioris, which I already mentioned. So 4 recti muscles, 2 oblique, and then 1 levatopalpebral superioris muscle. So what do they do? Functionally, they are divided into 2. Basically, one group of the muscle, they are responsible for the eye movement, while the other group is responsible for superior eyelid movement. Okay, the movement of the eye is being undertaken by the recti and the oblique muscle. So you have 6 muscles. So some people would say you have 6, muscle for eye movement, 6 extraocular muscles responsible for eye movement, which is the 4 recti muscles and the 4 oblique muscles. So this is a picture to show that, showing you the recti muscles and the oblique muscles. So you see the superior rectus, you see the superior oblique, the medial rectus, the lateral rectus, the inferior oblique, and inferior. So these are the 6. So now you can see, how they do their job, the different positions, as you can see, extorsion, intorsion, and all of that. So taking the eyeball, moving the eyeball into different positions, into as they are positioned. You can see the attachments. So the attachments, at the cornea will also show you that, where their movements is being undertaken. So whether it is to the right, to the left, extorsion, intorsion, and so on and so forth. So that's the basic function of these muscles, while the intraocular is basically responsible for um, accommodation and then constriction of the pupil. Then it's also good to note that levator is responsible for the eyelid movement, basically responsible for eyelid. I'll show that in the eyelid. You can see how it's positioned, at the anterior part of the eyelid, and then the superior part of the orbit, where you can have it, regulate the eyelid. Now, it's also important to mention that there is a facial muscle. This facial muscle is a sphincter-like muscle. It is also responsible for controlling the eyelid as well, because it arranges itself in a concentric manner in the upper and the lower eyelid. The major function is actually to close the eyelids, and also sometimes they assist with the tear drainage of the tears when they are coming. So this muscle is called the orbicularis oculi. Orbicularis oculi, why it's a facial muscle, it's a sphincter-like muscle, but it also has some level of role it plays in regulation of the eyelid. So far, what we've been able to achieve is to understand how the eye develops. So this is just a pictograph just to show you what we've been saying, all this while. We've been trying to, talk about a coronal section. We started from, the development of the neural folds, in the brain, and the appearance of the prosencephalon, and then further appearance of the diencephalon, which is a part of it, around the 4th week, and then the emergence of the optic sulcus, and then from there we talk about the emergence of the optic grooves. They grow into grooves, and as they grow into grooves, they're going further into vesicles, from optic vesicle to optic grooves, to grooves, from grooves to vesicle, from vesicle to cups and stalk, and then optic nerve emerges. So it all begins at this level, at the 4th week. Okay, so the surface ectoderm, parents of the surface ectoderm, neuroecta, and their mesenchyme. So this leads to several processes: appearance of the lens black coat, formation of the lens bead, and then up to the point we have formed the 2 parts of the optic curve, and then we have, the lens itself, we have the retina itself, and then we have the appearance of the optic nerve as a result of the fusion of the optic stalk, right? And then we have further appearance of the arteries, the hyaloid arteries, which eventually becomes the retinal arteries, which eventually even, degenerates. And then we have, the full blown, mature, eye having both the ciliary body, which are usually iris extensions of the surface ectoderm, and then or rather the optic curve, and then you have the choroid, you have other appearance of different other parts of the eye. This is just to show us a summary, but then let's look at where all of this gets to appear from. We established in the beginning that you're going to have the presence of the ectoderm and the mesodem contributing to the formation. So I just made a mnemonic to show you how to remember this, that what derives from what. So this is a mnemonic to show things that emerge from the surface ectoderm, things that emerge from the neuroectoderm and from the mesodem, and also from the neural crest. So you can take your time to look at them one after the other, surface ectoderm, neuroectoderm, and usually they majorly are sourced from the mesenchyme part of this layers. Now we look at, because this is an advanced course, we look at the molecular regulations that play a role in the control of the development of the eye. So I've been able to identify, this is an ongoing research, but we have already known molecules, or genes that are responsible for the regulation process, the developmental process. So we've been talking about induction of the optic vesicles, induction of the lens black code, and so on. These are being, done by specific, either sometimes you're going to see deleterious effects or mutations, what we call mutations, changes in gene action, or if we're having overexpression mutations, when genes are, overexpressive, the overexpression of those genes could lead to either one activity or the other. These are things that show us either get to manifest as malformations. They also get to manifest as different things. So one of them is PAS2, which is essential for optic stalk formation. Then you have RX gene, which is responsible for the spatial patterning and the morphogenesis of the optic vesicle. And then you have sonic hedgehog, very popular gene, and this divides the eye field, and then from which you have the appearance of the optic vesicles. So it divides the eye field, via the division of the forebrain, and also regulates the expression of even PAS2 itself. Okay, then you have the PAS-X, which is expressed at the anterior neural plate, indicating the eye field as well. So these are just indicators, more like things that show that they somewhat has some level of role to play. For example, the PAS2, if there is a problem with PAS2, you discover that there will not be retinal fissure. Remember the choroid fissure that has to, fuse, it has to close for there to be the appearance of the form, even optic nerve to form, and even for the hyaloid artery to give way for the hyaloid canal to appear. So if this doesn't form, in fact, for the retention or even of the vitreous body, okay, if this doesn't happen, you see that you have emergence of congenital malformations of the eye as well. Take a look at some of the clinical significance of the development of the eye. So you have emergence of several disorders that can occur as a result of development. One of them is, let me start with the ones that are not here. Aniridia, for example. Aniridia is the absence of the iris. Okay, so remember that iris is a structure, muscular in nature. It has some palpebral muscles, or that helps it to open and close the pupil to allow the light into the eyes. So you have the absence of iris sometimes, so we say it is aniridia. So most times when the development goes wrong, this usually can happen. And then you have detached retina. Sometimes the retina can detach. It can also happen as a result of accident as well. Can also happen as a form of congenital malformation. Impact in the eye can lead to this as well. especially during impact, growing up, and sometimes even as an adult, this can also happen as well. Then you also have what we call the coloboma. You also have the coloboma. Coloboma is a hole in one of the structures. It could happen at the iris as well, or the retina, or even the choroid. Okay, so it can cause a gap, usually causes a gap, and then could lead to non-separation. There is no clear distinction. For example, if it happens in the pupil or in the retina, you're going to have non-separation between the retina and the iris, and the cornea as well. Okay, so we have the anophthalmia. Anophthalmia is a form of a syndrome, usually the absence of one or both of the eyes. So sometimes you have the ocular tissues missing from the orbit, or even have the entire eye not being present as a result of maldevelopment process of the eyes, and this can lead to anophthalmia. Sometimes it comes as a syndrome, usually related to other forms of malformation. For example, the neural fold fusion, and also you have presence of short eyelids accompanying it, or even reduced eyelids accompanying it. Then we have what we call the aphakia. Aphakia is the absence of the lens of the eye due to surgical removal, and usually in cataract surgery this happens as well, but this can also happen even congenitally. It can come as a result of congenital anomaly as well. Okay, so it leads to the ability of the eyes to actually maintain focus, and then inability to look on a far sight or even look deeply in anything, because there is the absence of the lens of the eye because of the functions of the lens of the eye. So there is also the cryptophthalmos. Cryptophthalmos is a total absence of the eyelid, okay, due to either fusion with the skin of the forehead, or you're seeing the skin of the forehead being continuous with that of the cheek. So this usually, you have the eyeball being concealed by the skin, which is stretched over the orbital cavity, causing this. So you can say, I don't know. Okay, here basically what we have here is shown is the anophthalmia when one of the eyes is not present. That's the picture that is here. And then you also have the cataract. This is quite popular. It's a common disorder amongst us, usually as part of aging. So you have cloudiness of the areas of the lens of the eye. Of course, the lens is responsible for the, helping the light, us focus on light. Then when cataract comes, it comes, as a result of damage, to this cataract. Sometimes it comes as a side effect, of several disorders, for example, diabetes, and can also occur as a result of that. Then we also have sometimes you have persistence of hyaloid artery. The hyaloid artery, instead of canalizing, turning itself to a canal or degenerating, you still have it being retained and persistent. You also have that sometimes it could also be, it may not be problematic or lead to any particular dysfunction, but it's important to know that this can also persist as well. Thank you very much for going through the lesson. So if you have any question, you can reach out to me through my LinkedIn, my Twitter, ResearchGate. You can also hit me on an email. Yeah, thank you. I'll be looking forward to answering your questions.